Greetings, my name is David Walker. I'm a soybean breeder with the USDA ARS in Urbana, and I'll be talking to you about soybean germplasm with Adam Mahan, who's the current curator of the USDA soybean germplasm collection here. So soybean developed into a major commodity from a much less important forage crop in the early 20th century. Up until about 1936, soybean was grown mostly as a forage crop, and prior to the 1940s, most of the soybean varieties that were grown by farmers in the US were from the USDA collection, and they had been collected in East Asia, primarily in China, Korea, and Japan. By the late 1940s, cultivars had been developed from deliberate crossing of the most promising germplasm accessions that had been imported from Asia and other parts of the world, and intense selection to improve yield and to avoid traits like lodging and pod shattering resulted in a very narrow germplasm base. And so that's been a problem that's been difficult to deal with, but part of the value of the USDA soybean collection is to try and increase that germplasm base. So in this table, you can see that soybean acreage increased tremendously between the early 1900s and the mid 1900s. And so initially it was grown primarily in the southeastern United States and the eastern seaboard, and gradually it became more of a crop for the Midwest. In the 1920s and 30s, some people like Henry Ford realized the value of soybean seed as an oil source, and so we began to import soybean from northeastern China, but the Japanese invasion of that part of China in the late 1930s caused a problem with the imports, and that fed the need for more domestic production. And with the beginning of World War II, there was a much larger market for soybean oil in the United States. These tables show the development of or improvement in soybean yields between the 1920s and 2008. So this was a study done by Brian Deers, the soybean breeder with the University of Illinois, and his grad student Keith Rinker, in collaboration with researchers from the USDA ARS and other universities. So what these tables show are the yields of soybean varieties that were released during that period of time between the 1920s and 2008, all grown in the same environments in 2010 and 2011. So you can see that as time went on, the varieties had a greater yield potential. The top table is for maturity group 2, the middle for maturity group 3, and the bottom for maturity group 4. Around 1970, there was more of an increase in the rate of yield improvement, and that seems to coincide with the Plant Variety Protection Act and the ability of private companies to protect the varieties that they develop. And with their greater resources, they were able to accelerate the rate of yield improvement. So besides soybean genetics and breeding, a number of factors help to make soybean a major commodity crop. One is a wide range of maturity groups. So soybean can be grown from Canada down to Florida, and there are even some types adapted to the tropics. An important development was the mass production of combines and planters and other equipment specifically designed to handle soybean production and to enable it to be grown on a larger scale. Also, the development of crushing facilities and collection points in the Midwest helped to increase the amount that was grown in Illinois and the surrounding states. And as I mentioned before, Henry Ford was a very public and outspoken supporter of soybean use and finding new ways to use the oil and to improve the farming techniques used to produce soybean. As I mentioned earlier, the Japanese invasion of Manchuria in the 1930s disrupted a major source of soybean seed that was being imported into the United States. So with that need for alternative seed sources, there was a great increase in production in the United States, especially after the start of World War II. Chemists in the United States and Germany in particular, also at that same period, were working on ways to stabilize the oil and the oil components so that it would have a longer shelf life and be useful for a wider variety of commercial uses and industrial uses, as well as food. And among these would be the use of soybean oil in paints, plastics, cosmetics, and many other products. So after people realized that there was also a lot of protein in the soybean meal that was being separated from the oil, there was also a great demand for soybean meal and the use of poultry feed and other types of livestock feed. We'll soon be showing you a series of slides about soybean varieties that were important in the historical development of soybean in the United States. But I'd like to explain a few definitions and terms that you'll see in those slides. So when we talk about a germplasm accession or plant introduction, we're talking about a strain or variety of soybean that's cataloged and maintained in the USDA collection in Urbana. Most of the soybean accessions that are in the collection originate from either East or Southeast Asia, from countries like China, Korea, Japan, Vietnam, and so forth. But we also have accessions in the collection from Europe and the United States, some breeding lines and cultivars that were developed previously, as well as wild annual soybean and wild perennial species from Australia. And you'll be hearing a little bit more about those later. When we talk about maturity group, we're talking about a classification system that indicates how soybean or when soybean will flower based on changes in the photoperiod during the summer growing season. In Illinois, maturity groups 2, 3, and 4 are grown. The lower maturity groups from 000 to 1 would be grown farther north, and maturity groups 5 to 10 would be grown farther south. These are all increased, or the seed are increased to provide people requesting those from the collection. Maturity groups 000 to 4 are increased here in Urbana. Maturity groups 5 to 8 are increased in Stoneville, Mississippi, and maturity groups 9 and 10 are grown in Costa Rica. This diagram shows the approximate latitudes that maturity groups 000 through 9 and even 10 are grown at in North America. And again, you can see that 2s, 3s, and 4s are grown in Illinois. Before we launch into the slideshow, I'd like to make a few comments about these historically important varieties that we'll be showing you. The plants represent some of the most important varieties that were grown in the United States in the 20th century, and going from early introductions that were brought directly from Asia and selected for growth in the US just because they yielded well here, up into some more modern varieties that were developed in the 1970s and 80s and 90s. In each of the photos, the row that you can see on the right side that's usually partially visible is Dwight, a modern cultivar. So you can compare the row that we're featuring in the middle of the photograph with Dwight on the right side. The varieties and lines whose name appear in the text in bold are in some other part of the slideshow. And in the early 1900s, germplasm accessions in the USDA collection were often heterogeneous, that is, they weren't genetically uniform. So at present, they've been selected or purified so that they should, each accession in the collection should be genetically uniform. But because this was done in the 1970s and 80s, we can't be certain that varieties that were grown in the 1920s or 30s, for example, were exactly identical to what we have in the collection now, but at least they're closely related and similar if they are not identical. And the next slide, I believe, will be the beginning of the slideshow.
Hello there. My name is Adam Mahan. I am the new curator of the USDA Soybean Germplasm Collection here in Urbana, Illinois, and I am 2 months on the job. Hopefully, you all enjoyed the slideshow of the historically important soybean varieties. And then I'm just going to touch briefly on the germplasm collection itself. As you can see, we have in total over 21,000 different types of soybean that we manage and distribute worldwide each year. We average about 25,000 packets of seed that we send out each and every year to public and private researchers. The soybean that most everybody knows in Illinois is introduced the G Max there, Glycine Max. We actually have about 17,500 introductions. And so most of those are going to be soybeans that were collected or that were sent here from China or Taiwan or wherever it may be. Sticking with Glycine Max, we have the germplasm releases. There's 212 of those. Most of those are from USDA breeders, public breeders that just submit lines that everyone can use. We have the modern cultivars like the Dwight that was mentioned. We have something called old cultivars, which those would be more ancestor type lines. The private cultivars, we're up to about almost 700 of those. Those are going to be most of your soybean lines that come off patent. Once their 20-year plant variety protection certificate expires, we actually get seed of those and we can increase those and distribute those. And then we have something called ISO lines. So this would be, for example, you take Dwight and then you change one gene, typically an obvious change. It could be for height or seed color or whatever. And then now you have the ISO line of Dwight. We keep about 600 of those. We have genetic types. So these would be typically mutants for different traits that we keep in case anyone would like to use those for research. And then specifically for mutants, we have pigment mutants. And so these are seed color, blue, green, pretty much whatever you could think of we have. And then I skipped over it, but we also have something called Glycine soja. And that is actually the wild relative of soybean, of Glycine Max. We have almost 1,200 of those. Part of the collection we also keep, so soybean, there's Glycine max, which I talked about, and Glycine soja, which is the wild relative. We also have perennial soybean. And you can see there's lots of different types. We have about 1,000 of those total. And I did not mention this, but Glycine soja can actually be crossed with soybean, and you will get seed that you can plant and that you can use for breeding. Perennial glycine, you cannot simply cross it with regular soybean and get seed. But there has been some research done, a lot of research done here at the University of Illinois and with the USDA, where they actually took the Tomentella there, where there's 299 of them in our collection. They've actually been crossing Tomentella with regular soybean, and have been able to get some progeny from that. And there's actually quite a bit of research centered on what portions of the Tomentella genetics have crossed over, and still trying to figure out exactly what's at work there. But there have been some progeny that have yielded a high, and it's a pretty exciting area. And it just goes to show how the germplasm collection can be exploited. So then just a quick example of seed size, shape, and color to just give you an idea of the type of genetic diversity, the variation that can exist or that does exist in the USDA germplasm collection. The tiny black seed in the upper left-hand corner is going to be most similar to the Glycine soja, but probably even smaller than that. And a lot of times those tiny black seeds, they don't take up water very well for when you plant them and you want them to sprout. So a lot of times we have to do what's called scarify, where we actually rough up the seed coat to get them to germinate and to grow for us. Here's a few pictures of the Glycine soja, the wild species. And it is actually, if you can tell here, it's actually, you can't see the van very well. It's in a screen house. And the reason for that is the leaf hoppers and aphids and other flying insects, they really give these plants a hard time. And so we grow them in the screen house, planted in the field, but then we put a screen house over top. And you can see they're very viney. They do not really look like soybean. And again, we maintain about 1,200 of these, so you can imagine it's a good amount of work when it comes time to increase these. And then our perennial soybeans, they are actually exclusively grown in the greenhouse because they can take quite a long time to set pods and produce seed. Then just briefly, I wanted to discuss the history, at least the more modern history of the USDA germplasm collection. Dr. Richard Bernard, Dick Bernard as he went by, he was one of the most successful and highly respected soybean breeders from the United States. He developed several of the most widely grown Midwestern cultivars in the 60s through the 80s. These include Wayne, Williams, and Williams 82. And even after his retirement, he continued to breed vegetable soybean, known as edamame, as a hobby until he was in his mid-80s. Stuck around for quite a long time after retirement and really helped out his predecessor, who was Randy Nelson. So Randy started in researching soybean and soybean germplasm in 1978 and retired in 2016. He was an official curator of the germplasm collection from 1988 to 2016. The number of germplasm accessions, which I talked about, is up over 21,000 now. During Randy's tenure, that number increased by 45% to the current 21,000 that it is at now. On top of that, on top of maintaining the germplasm collection, he also conducted a highly successful breeding and research program to not only evaluate the soybean germplasm, but also to develop high-yielding breeding lines using that germplasm, using that diversity to contain unique exotic genes, trying to help broaden the relatively narrow commercial suite of germplasm for soybean. And then up to present day, here's Clint, Eric, and Todd. They help do all of the work that goes into maintaining the germplasm collection. Clint and Eric do most of the field work. And if you ever request seed from the germplasm collection, Todd likely fills those packets. As I mentioned, we send out over 25,000 packets each year. It keeps Todd very busy, not just filling the seed, but he also works with the international requesters, getting phytosanitary certificates, import permits, all those things necessary to ship from the US to other countries. And then last but not least, we have a picture there of David Walker on the left, myself in the middle, and Esther Peregrine on the right. Now, Esther, for the past 3 years plus, with Randy Nelson's retirement, has really helped take charge, and she really has been the curator of the collection. And if it wasn't for her, lots of small detail type things I think would have slipped through the cracks. So that is all we have for the presentation. Please feel free to email any of us if you have questions, or to stop on out someday and see the germplasm collection. Thank you for your time.